Okay, the epidermis is an example of stratified squamous epithelium. But let's have a closer look at what exactly is happening as we go from below upwards. Okay, the first layer is known as stratum basalis, or it is also known as stratum germinativum. Okay, now if you were to describe every layer or the cell of every layer, how would you describe it? Whenever in the practical viva anybody asks you to describe a cell, you need to have a pro forma at the back of your head. First, start by talking about the shape of the cell. Okay, please write these things down. How to describe a cell? Point number one, shape. Is it pyramidal? Is it circular? Is it cuboidal? Is it flat? Okay. Next, we talk about the nucleus and then the cytoplasm. So when you're speaking about the nucleus, there's only two things you can do. You can either say it's a hyperchromatic nucleus or a hypochromatic nucleus. If it's a hyperchromatic nucleus, that means that it is not an actively synthesizing cell because the nucleus has DNA inside it. And every time there is an active production of something inside the cell, parts of the DNA have to unfold, and whenever the DNA unfolds, it leads to a slight paler color, hence the term open face nucleus or euchromatic nucleus. If it's a cell that is not actively synthesizing anything, the nucleus is going to be nice and dark, and that is known as heterochromatic nucleus. Okay, now a couple of examples that come to mind are a cartwheel nucleus. Do you know where you see a cartwheel nucleus? It's seen in what is known as a plasma cell. Now, a plasma cell is essentially an activated B lymphocyte that creates certain proteins that are known as immunoglobulins, which we commonly call antibodies. So in the example of a plasma cell, certain parts of the DNA are unwound and certain parts are wound, because of which it gives to this characteristic spoke wheel appearance, and thus that is a characteristic nucleus of a plasma cell, a cartwheel nucleus. But moving back over here, to describe a cell, if you were to describe the cell of stratum basalis, just say 1, it is tall. You can say columnar as well. 2, the nucleus is euchromatic because these are actively multiplying cells. So the nucleus will always be an open face nucleus, right? A actively secreting and synthesizing cell or an actively multiplying cell usually shows an open face nucleus. Next, speak about the cytoplasm. Now, the cytoplasm usually has basophilic granules. In fact, here you can see that most of the granules over here are actually melanin pigments that are created by another cell called melanocyte and absorbed by these cells. So tall columnar, open face nucleus, basophilic cytoplasm showing some melanin pigment granules. That's the typical description of stratum basalis. If we move on, this little word here already reveals so much about the cell. Stratum spinosum. The cell is known as a spinous cell because the cells of stratum spinosum are attached to each other by desmosomes or tight junctions, but during the process of tissue processing, because of dehydration, the cell shrinks in size except at the points where the tight junctions have attached to each other, thus giving it a typical spinous appearance. Hence the name stratum spinosum. As we move upward to the third layer, this is known as stratum granulosum. Now, stratum granulosum, because it has certain granules which you can see all around the cell. Okay, now check out the shape of this cell. It's not tall, it's slightly more polyhedral, and all around the nucleus you can see these keratohyalin granules, and when you move upwards, certain changes start occurring in the cells. Now the nucleus starts becoming smaller and smaller and eventually becomes pycnotic. Okay, please read about what a pycnotic nucleus is. And eventually the nucleus completely disappears because the final layer is acellular. It's just flakes of keratin. And how is this keratin formed? It is formed because these keratohyalin granules are exocytosed and keratin fibers line themselves across the outside surface. Okay, so that's your stratum lucidum and stratum corneum. So if you observe, there is a change in the morphology of the cell as you go from base to the apex area. and every little change in the cell is reflective of what exactly is happening physiologically inside the cell. Okay, so that is important. Okay, I hope you've written down how to describe a cell. Talk about the shape, talk about the nucleus, talk about the cytoplasm. Is it basophilic? Is it acidophilic? Speak about granules. Sometimes you might have to speak about certain organelles that are more in abundance in certain cells. For example, a cell where there is active transport happening would be having a lot of mitochondria because you need energy for the active processes. Okay, more on that later. So let's move on to the next slide. Okay, now the transverse section of a blood vessel. What you can see here is an artery. This is actually a medium-sized muscular artery, and what you can see here is a vein. Now everybody knows the difference between artery and vein. There's a table, go through it. I'm not going to speak about all those basic things in the lecture. What I want to talk to you about is how to describe a vessel. The best way to do it is by going within outwards, right? So you need to start layer by layer. Always remember, start with the endothelium. When you talk about the endothelium, same principle. It's a flat squamous cell. Okay, the nucleus can be seen slightly bulging out, and the reason for this almost always is because an endothelium is not a simple cell where only exchange is taking place. It's slightly more complicated than that. So I want you to go and read about all the properties or characteristic features of endothelium. Note that down. Okay, there's a synthesis of so many prothrombotic and antithrombotic factors, like von Willebrand factor, for example, factor 8, I think, if I'm not mistaken. There are so many other receptors that are expressed in an endothelium which help in the migration of the cells from the intravascular to the extravascular compartment. For example, in case of an inflammatory response, if a leukocyte has to travel from within the lumen to the site of inflammation, then the endothelium has the responsibility of expressing receptors so that the leukocytes adhere to that and then they're transported outside. So the endothelium is not just any other flat squamous cell, but it's a specialized cell. So whenever you get the slide of a blood vessel, your viva almost always starts with enumerate the properties of endothelium. So read about endothelium. Then you've got the subendothelial connective tissue, which is like a thin layer of lamina propria. Always remember, underneath a lining epithelium, you almost always have loose areolar connective tissue. Almost always, right? So that's just going to be a couple of loose fibers lying around. And then you have tunica media, and the tunica media consists of, in this case, predominantly smooth muscle fibers. In case of an elastic artery like the aorta, there is more elastin fibers, and in case of a muscular artery, there is very less elastin fibers, which means that as you go distal from the heart, the elasticity reduces, and that's got everything to do with what is known as elastic recoil. So you read about elastic recoil in physiology. That's why you'll understand that the proximal you are to the heart, the more are the elastin fibers. The distal you go, the muscle fibers, the smooth muscle fibers start increasing. Now these smooth muscle fibers again are having some special features which have a great role to play in what is known as angiogenesis and the formation of atheromatous plaques. Read about it. Okay, tunica media of a vessel wall, a topic that would be really trending because atheromatous plaques is one of the most important pathophysiological changes that leads to coronary heart disease, right? So it's an examiner's favorite. And of course talk about the internal elastic lamina, which is more prominent in case of the muscular arteries. And then finally you have the tunica adventitia, which is predominantly collagen fibers, and nervi vasorum and vasa vasorum. Okay, read about all these things. When you're describing a vein, you already know the differences. The media is not going to be as prominent and as thick, and the adventitia is going to be a little more thicker in case of the vein, right? A few important things that they really ask about is what are post-capillary venules? Write that down. What are high endothelial venules? Now this is a good one. Please read about this. High endothelial venules. Then they always ask you about capillaries. So one of the most common questions is fenestrated capillaries and sinusoids. Okay, enumerate the sites where there are fenestrated capillaries. One of the most common examples is endocrine glands, and enumerate organs where there are sinusoids. Now one of the most important examples of sinusoids is liver, the hepatic sinusoids. So read a little more about these alterations in capillaries, right? Also read about the pericyte, pericyte. It's an important cell that they ask about. And some other things like arteriovenous shunts. Okay, right? So I'm going to move on to the next slide now. Okay, so now I'm going to talk about 4 examples where structure and functional correlation can be used very well. The first is a cell I'm going to describe, which is known as the proximal convoluted tubule, the cell of the proximal convoluted tubule. And it would be important that you know what exactly happens here. So I hope you've read renal physiology. The next thing I'm going to speak about is an exocrine gland. I'm just going to tell you the structure, a typical structure of an exocrine gland, and then just slightly differentiate between the acinus of a mucus and a serous salivary gland. Then we'll speak a little bit about the schema of the gastrointestinal tract in general. Again, not a lot of details. Then finally, we'll speak about an endocrine gland, and the example I've taken is the islets of Langerhans in the pancreas. Okay. Now, let's talk about the proximal convoluted tubule. You know that a lot of urine that is collected in the Bowman's capsule eventually is reabsorbed in the proximal convoluted tubule, right? And these processes, sodium, glucose, certain amino acids which are reabsorbed, are all active processes. Now this is what a proximal convoluted tubule looks like. Okay, and if you take one of these cells out, over here, this is the electron microscopic diagram of a proximal convoluted tubule. Let's discuss as we always do. Point number 1, shape. You'd say it's columnar, or you'd say it's tall cuboidal, right? Next, nucleus. Can you see? There's not a lot of black here, so it's an euchromatic nucleus, which means it's an active cell. And if you observe, you have basal striations, that is the basal aspect of the cell has gone into folds. Now just like in an enterocyte you've got apical folds, microvilli, which you obviously see here as well. Can you see them? These are all microvilli. This is more of a 3-dimensional part, okay? So just concentrate on the apical aspect over here. So these are the microvilli at the apical side, and these are the basal enfoldings. And all these black long things are actually mitochondria. So a lot of active exchanges are taking place along the basal aspect of the proximal convoluted tubule cell. Hence you need so many mitochondria, right? So similarly, if you want to speak about maybe the hepatocyte, you'd speak about how the hepatocyte has phagolysosomes, because detoxification is one of the important functions in a hepatocyte. How the hepatocyte has smooth endoplasmic reticulum, because lipid metabolism chiefly occurs in hepatocytes. So try to understand that biochemical processes which take place in organelles would be more predominant in those type of cells where these processes occur. Another example I can think about is just think about the interstitial cells of Leydig. The interstitial cells of Leydig will be having a lot of smooth endoplasmic reticulum. Why? Because what they secrete, that is testosterone, is a steroid hormone. If you have a cell that is secreting a steroid hormone, of course you're going to have the organelle where lipid synthesis takes place and lipid metabolism takes place. That's your smooth endoplasmic reticulum. So learn to apply your cells like that. Think about a cell and try to understand that the reason why certain organelles are more in these cells is directly pointing out to the function that is occurring across the cell. Do you get that? So what the examiner really expects from you is, has this person understood what to remember in this cell? And the moment you say that answer, they get really happy because they know that you're applying yourselves, and that's what it's all about. Let's go to the next slide. Okay, now salivary glands. Again, you know that glands can either be predominantly serous like the parotid, predominantly mucous like the sublingual, or they can be mixed like the submandibular gland. Right? The most commonly asked question here is the difference between a serous and a mucous acinus, which I will get back to. But eventually this is what you are used to looking at, right? This is what you think of when you look at a salivary gland. This is what you draw. Let's just change the perspective a little and see what it looks like in a three-dimensional format. Understanding this might probably give you a better perspective about what exactly is happening when a section is cut like that. Okay, so whenever you think of a salivary gland, or for any gland for that matter, I want you to think of a bunch of grapes. The grape is the secretory end piece. Like the name suggests, it's going to secrete something, so automatically the epithelium is going to become pyramidal or tall columnar. Okay, and eventually these secretory end piece secrete into the lumen, and the lumen eventually leads to the ductular system, and you can think of the ductular system as the brown twigs on which the grapes are attached. Okay, see what's happening here? The height of the epithelium is now reducing as you go from the secretory end piece towards the duct. Sometimes you have an intermediate stage which is known as a centroacinar cell. It's commonly seen more in the pancreatic acini. But anyway, tall columnar epithelium, now you've got cuboidal epithelium. As the multiple ducts join to form a larger duct, let's call this like an interlobar duct, then can you see the layers are increased. Now this is two layers of cuboidal epithelium, and this is actually an intralobular, but as it becomes more and more bigger, as more secretory end pieces and more interlobular ducts join, it becomes an interlobar duct, which will have 3 or 4 layers of not only cuboidal but columnar epithelium, right? And you see all these things, these are all the connective tissue fibers, and see how they intertwine like that. Okay, and look at that, that's actually another intertwining cell, which is actually a myoepithelial cell. When the myoepithelial cells contract, the grape will squeeze and the grape juice will come out into the duct. Okay, so this is what a 3-dimensional representation looks like, but in 2 dimensions, this is what you see. So of course, the darkly stained things are going to be sections that are taken like this. Why darkly stained? Because there are granules. Now these granules are going to be stained. Okay, if they're going to be basophilic granules, they'll be staining more purplish. If they're going to be acidophilic granules, they're going to be staining more dark pink, but they're definitely going to give a darker color, which is why the best way to differentiate between a secretory end piece and a duct is that a duct is going to be much more fainter. There is no granules in the cuboidal epithelium of the ducts. Okay, that's how you differentiate between an acinus and a duct, the epithelium as well as the color. Moving on, surrounding all these is going to be your connective tissue fibers, which you see here in light green, but they'll be obviously seen as little strands all around. Okay, it's mostly going to be collagen in case of exocrine glands. Then this is a larger duct. Now this is a larger duct, so you can see multiple layers here. Do you see that? This is a blood vessel. Okay, so if you see in this section, a myoepithelial cell just occurs as a little extra cell somewhere in a corner, just flattened. Okay, these are also known as stellate cells because they are kind of stellate in appearance, and they just surround the entire secretory end piece in this format. Okay, now once you understand what exactly is happening, let's move on to the differences in high power between a serous and a mucinous acinus. Very commonly asked question. Right now, this is parotid salivary gland. You can see all these dark granules. They're on the apical side. Look at how the shape is. Okay, it's pyramidal in shape. You've got a nice open nucleus because, of course, actively synthesizing things. These granules are essentially formed because there is active protein synthesis happening. Correct? So this is what a typical serous acinus looks like, and you can see 2 myoepithelial cells in the background. This is what a typical intercalated duct looks like. This is a striated duct. Now, remember we spoke about basal striations in the proximal convoluted tubule? Similarly, even in the striated duct of a salivary gland, there are basal infoldings and there are mitochondria over there. This is to increase the surface area, and the fact that there is mitochondria means there are certain things that are being exchanged even in the duct. Usually exchanges do not happen in a duct, but this is one of those exceptions, the striated duct of the salivary gland, where exchanges take place. Certain ions like potassium, certain immunoglobulins and lysozyme is actually exchanged in the duct here. Okay, As opposed to this, look at what a mucinous acinus looks like. It's light in color, and this is because mucopolysaccharides are lighter in color, and most of the times they are lost in the process of tissue processing. And see what's happening to the nucleus. It's flattened and it's at the base. Look, this is a circular nucleus because these granules are small and darkly staining. On the other hand, these granules are huge, almost transparent, and they push the nucleus to the base. Hence, a mucinous acinus has these flattened nuclei which are supposed to be drawn at the base. So many journal corrections. So many journals have been returned to people because they haven't got this they haven't gotten this right. So please get it right when it's asked in the exam, because the teacher will actually tell you to draw that. Okay. Now this is a serous demilume. In certain mucinous acini, there is a slight element of serous secretions. They just help in adding a certain liquidy feel to the otherwise viscous mucus secretions. That is the function of a serous demilume. You see that? Everything else pretty much remains the same. So in salivary glands, some of the most common questions asked are: differentiate between serous and mucus acinus, talk about the myoepithelial cells, describe in detail the duct system, speak about the intercalated and the striated ducts, and of course another question they commonly ask is the similarities and the differences between a serous salivary gland and pancreas. Okay, now in pancreas you're going to have certain light islands which are known as islets of Langerhans, so that you won't see in a parotid gland. On the other hand, in the parotid gland there is more connective tissue as compared to the pancreas. Okay, right, there's a couple more differences that you should really study. Okay, differences between serous salivary gland and the parotid gland.